right, hello everybody. We're going to be talking about cell cycle control. We already talked about the cell cycle in our mitosis unit, mitosis notes. Now we're going to be talking about controlling the cell cycle and then what happens when the cell cycle is not controlled, cancer. So I think these are super interesting notes, and I'll tell you what I think you need to know and what you just might be interested in knowing, but don't necessarily need to know for the College Board to what they're going to expect you to know on a test. All right, so, and I will mention that I am talking kind of quietly because my husband is napping. I'm sitting in the kitchen right now, and my bird is sitting next to me, and he makes kind of funny noises sometimes. So, just if you hear them, that's what you're hearing, just so you know. All right, so the diagram here should be familiar. What you're looking at is the cell cycle. Remember, interphase is G1, S, and G2, and then that little sliver, it was a different size sliver in our other diagram, it doesn't matter, is mitosis. So normally we control how many times or at what times a cell divides. And that's a super important part of preventing diseases like cancer. The cell cycle consists of a series of checkpoints. Now, this is weird to even imagine. The cell is basically checking on itself and it's making sure that its DNA is in good shape, that it's growing properly, that the chromosomes are attached to the spindles correctly, and that's what determines whether the cell gets to continue forward during cell division. And if it fails those checkpoints, then the cell stops and it might enter G naught, it might go through apoptosis and die. So that's what we're going to be talking about, is just how that happens and then what happens when it doesn't happen correctly. So here's an example. At the G1 checkpoint, it determines if a cell should replicate its DNA. And from here, the cells either enter the S phase or they go on to G naught. And I'll talk a little bit more about the G1 checkpoint in just a second. Remember that the word senescence, we mentioned that in the mitosis notes, just refers to cells that have stopped dividing. And that's often they've stopped dividing because they're old. So think of the word senile. It's the same root word. And so just think old cells are going to stop dividing. Okay, so this diagram I stole directly from the Biozone ebook. I think it's got a lot of really helpful information. So it's got the cell cycle, and then it's got the G1 checkpoint. And these are the things it says about the G1 checkpoint. It's checking to make sure the cell is in fact large enough, that the cell has enough nutrients to divide, and that signals from other cells have been received. So it is in fact time for it to divide. Then there's a G2 checkpoint. This one is also checking to make sure the cell is in fact large enough to divide and that the chromosomes have been successfully duplicated. Remember that that's what's happening. DNA replication, I'm showing the bivalent chromosomes, are being made during the S phase. And if that gets screwed up somehow, the G2 checkpoint, what is a G2 checkpoint? It's a crazy idea. It has to do with enzymes and signals that are being received. It's a very crazy concept that it can check on itself. But anyway, if the cell has not correctly replicated its DNA, those mistakes need to be repaired, or the cell has to step out and no longer divide. And then the last checkpoint is the mitosis or metaphase checkpoint, the M checkpoint. And it can pass that checkpoint if all of the bivalent chromosomes are correctly attached to the mitotic spindle. What we don't want to have happen is the wrong number of chromosomes going into the two new cells. So you should have 46 chromosomes lined up on the in the middle of the metaphase plate, and then all 46 halves should go to the left side, and 46 monovalent or sister chromatids should go to the right side. You don't want extra chromosomes going one way or the other. That does happen on occasion, and that's a genetic problem. Okay, and then the last thing that I really like about this diagram is they're kind of going into G naught and just a little bit of information. And I wrote a few definitions up on the side for you. So when do cells enter G naught? Cells may exit the cell cycle in response to chemical cues, and those chemical cues might be coming from within the cell, which we're going to learn about, or coming from externally from cells that are around them, and that will determine whether they stop dividing. So cells in G naught might be quiescent, which basically means quiescent means quiet. They're waiting to be reactivated. They may be differentiated. And what that means is that they're fully functional cells. They're just never going to divide again. So, a great example of that is like a nerve cell. Nerve cells are working, they're working fine, but they don't go back through cell division again. Or the cells that are in GNOT might be senescent. So they're old, they're no longer dividing, they may have accrued damage in their DNA, their telomeres might be short. We're going to learn what telomeres are in just a minute. Okay, so let's take a look just to give you kind of background information on how the cell cycle can be controlled. So I said, how are they controlled? I'm telling you that they're controlled, but it's a weird concept. So it says, evidence from this classic experiment, I'll move my picture, suggests that there are molecules present during the cell cycle that cause cells to progress to the next phase. So let me get my picture out of the way. I've got two experiments going on. In the first experiment, the scientists took a cell that's in the S phase, so it's copied its DNA, and we've got a cell that's in G1, and it merged the two cells together. Immediately, the cell that was in G1 jumps to the S phase. And it says when a cell in the S phase was fused with a cell in G1, the G1 nucleus immediately entered the S phase and DNA was synthesized. So there must be a molecule present in the S cell that's triggering the nucleus that was in G1 to move on to the next stage. In experiment 2, this one's crazy. We have a cell that's in the M phase. It's ready for mitosis, and they fuse it with a G1. Immediately, both nuclei now are in the mitosis phase. However, the one that was in G1 never copied its DNA. It never went through the S phase or anything. It just jumped right to the M phase. And again, that's telling us there are certain molecules present that are triggering mitosis. And so as soon as those molecules are present, the cell goes into mitosis. So let's talk about how that's even possible. So we're going to get that here. So this is, well, I'll show you these diagrams and everything. Internal controls. So I mentioned that there are signals within the cell that are going to trigger mitosis or stop mitosis. And there are external controls, things from the outside. So this is an example of an internal control. Now, to be clear, the College Board says very clearly, you do not need to have this stuff memorized, this page. You don't have to have memorized. However, I have seen this on AP exams. I've seen this exact diagram on AP exams. So they're going to give you all the information. You don't have to memorize it, but you have to understand what's happening in it and then be able to solve or answer questions about it. So let me talk you through it really quickly so you feel like you know what's going on. So protein molecules are present in varying concentrations throughout the cycle, and that's what's going to determine what stage of the cycle we're in. So one example we have is called the mitosis promoting factor, or MPF. So I want you to take a look at the cycle down at the bottom, and then I'll move my picture so we can look at the graph above. So CDK is called a cyclin-dependent kinase. Now, if you remember from 2 notes ago, kinases are proteins that phosphorylate other molecules. So they add phosphates to other molecules. So cyclin-dependent kinase is present in a constant amount. So take a look at the cell cycle. Notice we've got M, G1, S, and G2. Look at CDK. It's the orange puzzle piece. That's an enzyme. It's a protein. Okay. CDK is activated by a molecule that's called cyclin. So look for cyclin. It pops up. It's purple. And as it accumulates, it starts triggering more and more of the CDK. It fits with the CDK. When the two are together, when they fit together, it now has a new name, and it's called MPF, mitosis promoting factor. So it's the cyclin and the CDK together. And once we have MPF in large enough quantities, concentrations, that triggers mitosis. So if we don't have MPF, we're not in mitosis. If we have a good concentration of MPF, the cell goes into mitosis. So that's an example of internal controls. So it says down at the bottom, it activates other proteins needed for mitosis, like microtubule formation, After mitosis is over, otherwise it would keep going on and on and on, the cyclin, look at the diagram, the purple gets broken apart. So now that's gone and it has to accumulate again. So over the course of the rest of interphase again, we start getting more and more and more cyclin building up, and it starts connecting with the CDK and making MPF. And so the cycle keeps happening again and again. If you take a look at this diagram here, the graph, I'm sorry, that's next to my picture here, you see mitosis, G1S, G2 mitosis, G1S, G2 mitosis, so forth and so on. And so you see the cyclin, that's the purple, you see that increasing over the cell life. And then once it gets into mitosis, then its level drops off because it got degraded. And then it increases again over time, and then it drops off. Notice the orange line, that's the MPF. So as more cyclin forms, it starts joining with the CDK and making more MPF, the mitosis promoting factor, and that triggers mitosis. And notice that both hit a peak during the M phase, and that's what's triggering mitosis. So that's an example of an internal control of the cell cycle. Oh, and I wrote down at the bottom, if there's no cyclin, then there's no mitosis. So stopping cyclin production tells the cell to go into G naught. If there's no cyclin, we're not going to get any cell cycle. We're not going to get any cell division. Okay, here's an example of things coming from the outside that are going to impact the cell. So this one refers to platelet-derived growth factor, PDGF. Again, you don't need to write this down. I just need you to know that there are internal and external factors that are going to impact it. So PDGF, platelets are things that are trigger blood clotting. So when there's been an injury, platelets get shunted to the area and they help the blood to clot. Well, it says, why would they release a growth factor? Well, they release a growth factor because there's been an injury in the area and we need cells to repair that damaged tissue. So it says the growth factor stimulates cells in the injured tissue to begin dividing to heal. And so I'll just show you this really quickly. You don't need to understand this whole diagram, but basically we've taken some tissue, we've cut it up, and we've just got some cells that are all by themselves. They're not connected to any other cells. There's no other anything going on. And if there's no platelet, no PDGF, if there's no growth factor, they just stay like they are and they're quiescent. They sit quietly in the G naught phase. But if you add PDGF to it, suddenly they start dividing. And I want you to notice this diagram. They divide until they fill the container and then they stop. And that's going to be our next external control. The next external control, give me a second to get my picture out of the way. No, not in the right place. Okay, it says protein and other environmental signals are generated by other cells. This next external control is called positional inhibition. I've also seen it called contact inhibition. So take a look at the diagram just so it makes sense. So you have in this scientific dish, like a petri dish, you have different patches of cells, and they're going to start dividing, and they divide until they touch each other, until they fill the dish, and then they stop. So it says here, normal cells must be anchored and not too crowded. They are density dependent. Once they sense, it's a weird thing, once they sense that they have filled the space, they stop dividing. So for example, if I skin my knee and the cells go through mitosis to repair the injury, so I have a big gap in my skin. The cells divide until they touch each other. And then they know, okay, positional inhibition, stop dividing, don't keep dividing. Well, here's the weird thing. Cancer cells don't have that. They don't have positional inhibition. So if you put cancer cells into a dish, not only do they fill the dish horizontally, but vertically also, and they pile on top of each other, and they form a tumor. That's what a tumor is too many cells piling on top of each other. They don't have positional inhibition. They lack positional inhibition. All right, so let's talk about what cancer is specifically. Cancer is uncontrolled cell division. I'm sure you figured this out. Let's go over a couple reminder things before we get into cancer itself. So first of all, we've mentioned apoptosis before. I'm going to bring it up kind of more detail right now. Apoptosis is a process of programmed cell death. And when I say programmed, I mean that it's programmed in the DNA for damaged cells before they can multiply. So the whole idea is that we're going to sacrifice, especially for cells of a multicellular organism, we're willing to sacrifice individual cells to protect the organism itself. So it says the cell is sacrificed to prevent the spreading of its mutations to new cells. So if I have a mutant cell and it goes through cell division, now I have 2 mutant cells. If they go through cell division, now I have 4 mutant cells, and so forth and so on. So apoptosis is supposed to stop this mutant cell from dividing. So what happens in apoptosis is that it shrinks, it blebs. Yes, scientists have a sense of humor. The little blobs are called blebs, and the DNA gets fragmented. And then the white blood cells, they just come along and they gobble up any cell that's gone through apoptosis. Done. It's brilliant. Mutations in cancer cells, fascinatingly enough, stop or inhibit apoptosis. So when they should be blebbing, they're not blebbing. Cancer is brilliant, and it's horrifying how brilliant cancer is. So this is due to failures in the cell cycle checkpoints. So the cell cycle checkpoints have mutations in them in a cancer cell. And so all those things that are supposed to prevent cancer, they've been mutated, and now we're developing cancer. So we often say that cancer cells are immortal. What do we mean by immortal? I can kill a cancer cell very easily. I can squash a cancer cell and it'll be dead. But cancer cells don't, number one, they don't kill themselves, and they keep dividing and they keep dividing and they keep dividing. We're going to talk about Henrietta Lacks at the end of these notes. Maybe some of you are familiar with HeLa cells. And her cells, they were cancer cells that were collected in 1951, I believe is the date. And those cells are growing in scientific laboratories all over the world to this day. Even though the woman who owned those cells passed away in 1951, her cells are still dividing because they're cancer cells. So they're immortal as long as they have the nutrients and the space they need in order to grow. Okay, the other thing that I want to talk about are carcinogens. Carcinogens are things that cause cancer, and we say they're agents that cause cancer because they're not necessarily chemicals. Usually we think of them as chemicals. But for example, ultraviolet radiation isn't a chemical, so we say that it's an agent capable of causing cancer. I'm going to have to pause my video because my birdie's getting loud. Jonah, are you okay? You might be okay. I'm going to keep going as long as I can. Okay, so carcinogens are mutagens. They damage DNA, and that disrupts the cell cycle. You don't need to write this down. I just want you to be aware of it. For those of you that are interested in medicine or cancer, it's a super interesting topic. Maybe you have someone in your life that has cancer. This is a super interesting article called 6 Common Carcinogens Everyone Should Know About. I'm going to go ahead and pause my video right now, and I'll finish up in just a moment.